All right. Hi, everybody. This is the lecture on enzymes, energy, and work. So I skipped ahead a few, or kind of went back depending on which ones you're looking at. So we could look at enzymes and how they actually accomplish this work chemically. So a lot of you are in chemistry, so this will actually help. So the intro slide was just, again, a good reminder of this sequence. The picture over here is going to do a really good job showing that DNA is always just going to stay in here. It can translate to RNA or transcribe, and then the RNA goes out of the nucleus. Ribosomes produce proteins from RNA. And everything that we're dealing with in this section of the class, they're always going to be proteins. Proteins are what provide all the function in the cell. This is a basic look at what an enzyme does. In this case, for example, we're fusing two things together. These can be chemicals, something like that. Normally, they're not going to want to both come together very well. It's supposed to be an arrow. And you would just have to wait in nature for these two things to hit each other, which can take a long time. So enzymes provide an active site. And remember, these are all just protein folds in these active sites right here and here. These are all just amino acids that fit perfectly. And that perfect fit at the active site causes things to come together. This is a very good first example of, I'm not going to draw it, but this, for example, could be ADP. I can't write that here. And this could be the little phosphate, the phosphate. So in that case, you would have, I don't know why, I don't even know what that thing is. But you could have an ADP right here, a phosphate. They get put together, and then the product is over here. So this could be a simplified version of your ATP synthase. But essentially, what enzymes are doing is bringing things together that normally aren't going to come together. This is important too. Basically, we're going to cover this, and we covered this in the other videos. Glycolysis goes to pyruvate. Pyruvate is converted to acetyl-CoA. That goes into the Krebs or citric acid cycle. Do not forget this step here. So if I ask you what the input of the citric acid cycle is, it's not technically pyruvate, because pyruvate has to go one more level into acetyl-CoA. That is your input for the Krebs cycle. So they're still directly connected to one another down here, but just have to remember this step right here. It's important because if you want to incorporate amino acids or fats, they are very easily and readily made into this acetyl-CoA molecule. And that is how they can kind of contribute to metabolism. Okay, so this is a really good look at pretty much everything that enzymes are doing. You have a substrate that, even in this case, maybe it wants to break apart, but it's not going to unless given enough activation energy to do so. What the active site in the protein allows is a place for it to bind. When it binds, the protein sometimes shifts or basically cracks a little bit, or like moves just a little bit, enough to make this crack right here. You split those in two, and then now you have a split product. Basically, the whole point of the active site is to provide the perfect environment for which something can bind and basically undergo a reaction that it normally wouldn't be able to. Best part is that the enzyme is renewed. You don't ever lose the enzyme by making more products. So I may ask you to draw this or do all kinds of stuff with this. Essentially, the concept that I want you to know is that although maybe two things, for example, so we're going to cover energy, although they may have an energy here, and a drop in energy is actually a favorable thing to happen. Remember that Gibbs free energy? If it's negative, that means it's a spontaneous reaction. It wants to happen. That doesn't mean that it's just going to happen naturally all the time. You need to provide this perfect active site for this to happen, so you get to here. When you do not have an enzyme, you have to muster up all kinds of energy, usually through heat, to get up to enough point, which, yeah, so the main point is up here. That's all that activation energy that you need to provide all the way up to here. before these two things can split apart, if this is what you want. With an enzyme, though, everything's fine. That reaction energy is a lot lower. It's going to be right here. And essentially, you get away with doing a lot less. So right here, all the way down to there, you don't need to provide all the energy that you normally would when you use an enzyme. Okay. So we get into these three terms. Exergonic or endergonic, Gibbs free energy, and, I would just say favorability. Okay. What I need you to know, I'm not going to give you this equation to do any math or anything, but it will definitely help your understanding if you know where Gibbs is coming from. The whole point of Gibbs free energy is that a negative value, I should do it down here. A negative value down here is going to mean that this energy or the reaction that you're measuring with this Gibbs, it's going to release energy. Energy released is negative energy right here. Okay, basically what energy is usable after the reaction occurs, and that negative energy is what's going to be released. So if you want to look over here, you can see that, changes in heat can manipulate this number, changes in entropy. So for example, a reaction that has higher entropy, so the one that we saw up here, this is a higher entropy reaction because one thing is going to become two. And remember, the universe wants to basically spread out infinitely. So that means that the energy in this case is going to be a favorable amount. And that's the case, and that's how you can remember that when we split bonds, we often end up with a negative energy, which is good. That negative energy means that you are releasing energy into the environment. We kind of highlight that down here. Oh, I can draw a star, nice. And same thing goes for breaking bonds. Or same thing goes for forming bonds, just opposite. You usually need to add a lot of energy to form those bonds. And when you form bonds, you end up with a positive Gibbs free energy. So it takes a lot more energy, get out of here. It takes a lot more energy to form a bond than it does to release one. You still need enzymes at each of those different steps for the most part so that we can control when these reactions happen, even if it's a negative good reaction. So you're going to have enzymes at all these steps. So definitely clarify with me if this is still unclear. We're definitely more in the realm of physics right now, but Gibbs is still a really good indicator, for example, if a reaction is going to move forward or if it's at least a little bit favorable versus no, it's not favorable because it's a plus and you really need to put in some energy to that. So we kind of talked about this already. If you have a reactant, like we saw in that first section, and it's going to go to more products and you're going to break some bonds, you're going to go down in Gibbs energy, and that energy is going to be released. So we introduced this word up here. When energy is released, that's exergonic. Okay. Oh, yeah, it's right here too. The reaction is what we call spontaneous, meaning that eventually, given infinite time, it's going to happen. These bonds will eventually break. So you still need to input some energy to get to this stage, though. So it's not just a completely an easy street. So in the case when you need to add energy, this is normally going to happen when you are forming bonds, just like right down here. This is what that positive Gibbs is going to see. See how that's positive in this case? That means you are going to need to add a whole lot of energy for this to happen all the way up to here. And then eventually your products, when they settle down, they'll end at a higher energy level than when you started. That energy has to come from somewhere. And in a lot of cases, we'll see that it comes from coupling reactions that are very favorable to ones that are not, like these endergonic reactions. You can think of these as non-favorable reactions. These need tons of energy to add. So it's a good way to remember photosynthesis is a process where we are adding bonds to things. We are making glucose, we are making ATP. We're not using any of those things. So all this energy that photosynthesis is working with, it's working with something that is trying to actively form bonds, which is very difficult. So, good way to look at it. If Gibbs is low, that means we're going to go. If Gibbs is high, you're just going to have to kind of wait around because nothing's actually going to go one way or the other, probably. You're going to have to input energy to change anything. Well, this is 0. This is a bad example. Just nothing's going to change then. So, another good little graphic. Basic thing is that when you break things down or catabolize things, they're exergonic. A lot of that time you're going to release energy. You're going to be able to form ATPs. ATPs go on because you formed a bond, releasing that energy sometimes. But if you want to do any work or any kind of changes to proteins that you need to make changes to, you may need to break that bond so that you can release some of that energy. So again, when we're looking at something, when you start with a big, massive carbohydrate, it's a higher energy reactant. As you break that down, some of that energy is going to be released. And in this case, that energy can be used to form products in the cell or in the organism that normally wouldn't go forward. And this is the endergonic reaction that's going to require that energy. So you gain energy by breaking things down. Here it is. Now you can use it to make complex systems happen, like forming bonds. Okay, so we looked at ATP. I've told you before there's 1 phosphate group, 2 phosphate group, 3 phosphate group. All this other stuff over here, it's not really ever gonna change. You don't have to worry about it. Biggest thing with these ATP is that this is a reaction that massively wants to happen. It's a very spontaneous reaction. So if this was a Gibbs energy, it would be negative. Let's put that there. So this isn't measuring it in Gibbs, it's measuring it in kilocalories per mole. That's the energy that we're releasing because this phosphate group is basically spring-loaded to get out of there. It's huge, it's charged up against these 2 repellent phosphates. It wants to, more than anything, escape and bind to something else instead of ATP. That's why forming this bond is so difficult, and that's why ATP synthase needs to rely on this that massive proton gradient, that hydrogen ion gradient. But as long as you have an ATP, you can use it in enzymes to move reactions forward that wouldn't happen. For example, you have a glucose and one of those phosphate groups. Okay. You need to get that phosphate group added onto glucose. This is not going to happen under the laws of nature if we do not intervene, basically. A positive net Gibbs energy means that you're going to have to input some kind of energy to get this done. The way around this is by taking another reaction that wants to happen, like ATP, splitting into this phosphate, for example. That really wants to happen a ton. And if you combine these two, you end up with a net reaction. See how these are now combined? So enzymes defy this. They defy the natural order of things because they combine reactions. They combine a very favorable reaction with an unfavorable one, because then your net reaction is going to be favorable. We kind of went over this with hexokinase, the enzyme. The fact is that hexokinase will only work when you have a glucose and an ATP to bind. They fit right here and right here. When they fit, both of them are going to be forced together. This reaction happens, and this reaction happens because they couple it so that this whole reaction all has to happen at once. That's the key thing, is that the only way an unfavorable reaction is going to happen is if you pair it up right away with another really good reaction. That's enzyme coupling. That's saying here's this terrible reaction, but if we make it happen at the same exact time that this really favorable reaction happens, both of them are still going to go. That's why ATP is so powerful, is that it's going to always provide this good negative Gibbs energy that's going to force all these bad reactions together. Because what you're doing here is adding a phosphate to glucose. It doesn't want to form a bond. It's fine how it is. But with the enzyme, you have a site where both things are going to come in contact. You're going to force that bond. That's the power of that enzyme. Equally, so we kind of touched on this separately in class, basically once you've formed enough product, and this is what we call feedback inhibition, when you've formed enough product, what it actually does is it basically says, we've basically formed enough product, it's going to bind to a site on the enzyme that will essentially shut it down. This is, you can view this as an allosteric inhibitor site. Now, an allosteric inhibitor in its pure form is a chemical drug inhibitor that maybe comes in here and that messes up the entire 3D structure of the enzyme, and it can never work again. In the case of feedback inhibition, it's different because although it's going to do the same thing to the structure and change it and turn it into an inactive form, it's only doing this because the product itself is saying, okay, there's enough product of me, we don't need any more, shut this whole thing down. And a big thing is, and this highlights it, is that you need to shut these glycolysis enzymes down a lot of the time because otherwise they're just going to keep burning your energy off, and that's super unhealthy. Okay, the reason this reaction has to occur, why we need to get that phosphate on in this first step of glycolysis, is that the minute you add this massive phosphate, this glucose cannot get out of the cell. It's stuck. That's perfect for us. We want to keep that glucose stuck in and do glycolysis. Number two is a little less intuitive. Basically, when you make a glucose into glucose 6-phosphate instead, you actually now don't have to worry about the 50-50 that glucose is still in. You know how we talk about gradients? We basically take this thing out of the gradient forever. It's no longer glucose. It forms its own 50-50 now across the membrane. It's kind of a goofy physics entropy style of thing to worry about. So if that makes sense to you, you can use it on, you know, like one of the optional questions maybe on the exam that you'll see. If not, it's not going to factor in like strictly if you don't like it. So we talked about glycolysis, and we'll obviously, you know, we have bigger review videos for this already. But for the most part, yeah, you're going to start with the glucose. You're going to stick it with these two phosphates. That's unstable. It splits down the middle. You end up with these three guys. They're basically the same thing. They'll convert to that. And then after that, you actually break a few bonds down, which releases good NADH, releases ATP twice, which is good, because now that happens twice. So you actually net a nice 4 ATP, and you end up with pyruvate. Now remember, this is going to go to, I'm not going to be able to spell. Oh boy, acetyl-CoA. That's what this is supposed to say, but it won't let me. Acetyl-CoA. And then the rest of these slides really are kind of more supplementary to what we've talked about. They're another visualization of something happening. Like in this case, you see that if you want to reform this bond over here, what the enzymes do with that active site is they form what's called a transition state with the molecules. Basically a state where all three molecules are bound at the same time, and the enzyme active site is the only chemical place that's going to fit that perfectly, so that ultimately the product spits out. So you still need to add a little bit of energy, and sometimes that can come from, you know, different sources. But what you want right here is that you get that beautiful transition state, everything's all locked in, you can break these bonds in this state because they're you kind of have wedged this bond. Remember, we're breaking this bond over here. You've kind of wedged that bond. In your transition state, though, this bond is actually a lot stronger. So then ultimately, this goes off together. So this reaction wouldn't just happen without this perfect 3D structure right here. So that's why interfering with enzymes and their structure can have the big bad consequences. This is another good supplemental slide, basically that ATP is the molecule that is going to be going in and out throughout your body and throughout respiration the whole time. It's either going to be formed by breaking down a glucose, you're going to form energy, you're going to form ATP. And we do that at the electron transport chain, a little bit in glucose. You can use that chemical energy to go and build large molecules, like small amino acids, can become massive proteins. we need energy to do that. We spend that energy, we repeat the process, and head back here. This is just another good example of, yeah, glucose is going to break things down. Glucose is going to get, metabolized. ATP comes out. ATP is the chemical energy that we use to facilitate reactions. Because remember, we want net exergonic downhill favorable reactions to happen. Because the energetics of those mean that they're physically, in physics, favorable. These are just good to show that these active sites, they're just going to form a perfect spot where bonds can be broken. Another good thing is that they can form bonds by having tight little pockets right here. That bond gets formed because this active site right here is the perfect setup for two molecules to come close together. Pretty similar to the other slide, just another way to look at it. I'm not going to strictly test you on a ton of reaction speed stuff. It's good for you to know if you want to elaborate on your answers. Basically that an enzyme can go and go and go as you add more substrate, but eventually you saturate the amount that the enzyme can do. If you have so much substrate over here, you eventually reach a point that the enzyme can only go so fast. This process can only keep going over and over so quickly that eventually you settle down. So, besides competitive inhibitors, allosteric inhibitors, you also sometimes have little organic molecules called coenzymes. A lot of the times these are vitamins, for example. So like vitamin B12 needs to bind to one of our brain enzymes. When it's bound, only then can the substrate fit. Whoa, that was going to happen. Only then is the substrate going to fit after you bind the vitamin. So you don't need to know the name apoenzyme. All apoenzyme really means is that it's an inactive enzyme. You need to bind this coenzyme right here before you can bind that substrate. All right, I think this is mainly just interesting stuff. Yeah, like how metabolomic pathways evolved. It's kind of interesting how they always evolved step by step. This is a really good one. This is like what it looks like in an active site. Because remember, amino acids have all these special groups. They have all the special bonds. They are perfectly aligned for only that substrate. If you change any of these amino acids and you change the little group that they have, like for example, that's a negative group right there. These are hydrogen bonds that are going to form. These are positive groups. See how the substrate negative goes to the positive, vice versa here. If you change any of these and you like erase this one and you mutate it or something, the whole thing can go down because you no longer have an active site. Equally bad, so not at the active site mutations, if you screw something up way back here with these proteins, it could have far-reaching consequences for the whole thing because the structure of the 3D protein is so incorporated even in these like scaffold proteins in here that I'm drawing in. I know this is a little messy. If you change even the structural proteins of an enzyme, like in the backbone, the whole thing can just unwind. So that's why mutations are usually so detrimental. This is your great slide. This pretty much is going to provide everything you're going to need if I ever ask you what is going to take down an enzyme. You have a lot of different answers here. One thing that's not in here is the little coenzymes. So one thing you could add to this slide is those little vitamin coenzymes. They can essentially turn on or off an enzyme, and so can kinases too. Sorry I didn't draw that, but we did go over them. So supplemental, but yeah, here's your competitive inhibitor. Here's your normal substrate. Competitive inhibitor blocks an active site. It's really easy, intuitive to remember because it competes with your substrate, and then the substrate never binds. An allosteric, a non-competitive inhibitor, or an allosteric one, I will label it as an allosteric one, by the way. So, because non-competitor is weird. So, an allosteric inhibitor is not going to actually bind to the active site. It's going to bind to somewhere completely different. And what that binding does is basically ruins a bunch of the structure in here, or changes the structure so that you lose your active site. So it's still going to take down the entire enzyme. It's only a matter of, it's not actually physically blocking the substrate. So remember that little changes in here can cause wide structural changes if something binds here. And remember that sometimes feedback inhibition, so like product that, enough product has been made, this is a lot of the time where product can bind. It has a secondary like on and off switch. Similarly, if you want to add like a P, I hate these, what is that thing? If you want to add like a phosphate, for example, remember we talked about kinases, if you add that, for example, maybe here sometimes, that's kind of functioning as a inhibitor as well. But when I use the word strictly non-competitive inhibitor, when you hear the word inhibitor itself, it usually means some kind of chemical structure that either we've designed or the body has to turn things off. I'm not going to ask you about speed, but again, if it helps your understanding better, better for you. So if you have a competitive inhibitor here, if you add enough substrate, you eventually, see this red line is the enzyme with competitive inhibitor, you eventually get back to your Vmax and nothing changes. You just have to add a lot more substrate. If you add an allosteric inhibitor though, you will change a lot of the enzymes for the worst. So much so that the Vmax in this red line this time, it's never really going to go back to the Vmax that you originally had, because you will literally just take down a bunch of your enzymes. So no matter how much substrate you add, tons of them just are not working at the same level as they used to be. So this is the slide that really covers feedback inhibition. If you have a substrate, it breaks down, everything's going well, it's a big old chain of stuff, you end up with this end product. That product itself is going to shut down enzyme number one. The whole thing goes down. So this is a really good way for cells to stop pathways that they don't need to use anymore. Once you get that end product, it's going to bind allosterically and inhibit this enzyme, pathway shuts down. And then we talked a little bit about phosphorylation, kind of a similar idea. If you add phosphate groups, it's going to change all the structure either in the active site or in the whole protein. See how these two, here I'll erase those. See how these two structures have slightly changed. Like this one's a little more open. It's kind of shifted things around here. See how this green loop is all the way over here after adding the phosphates. Over here it's kind of doing its own thing like that. In this case, when you add one of those big phosphates, and you remember you use ATP to do that, and you couple the reaction, is that you can actually activate something, make it go. So we covered this last class. Phosphatases take off power or everything. Kinases, they put them on. This can either turn proteins on or it can turn them off. It depends. But they are a good on and off switch. Okay.